Welcome to our review on osmosis. So what we've already looked at then is the idea of diffusion, and osmosis is very similar, just a little bit more specialised. So whenever we're talking about osmosis, the definition we've got to remember for this is that osmosis is the movement of water from an area of high water concentration to an area of low water concentration across a partially permeable membrane. Now normally you'll get 2 marks for that, one for that first section about moving from an area of high water concentration to an area of low water concentration, and the second mark is we're talking about the partially permeable membrane. So these particles are still moving down a concentration gradient, but remember it's only water that we're talking about with osmosis. Again, we're talking about a passive process, and when we mention this partially permeable membrane, we're referring to one that has these small pores in it that are only going to allow certain molecules through. So what we tend to find is in the diagram in the bottom left there, you can see that the pores are small so that the water can pass through, but the sugar is too big. So obviously partially permeable because sugar can't pass through it, but the water can. We need to understand what happens in terms of osmosis inside our plant cells first of all. So on the left hand side of the diagram you can see a situation where we've put a plant cell into a very dilute solution. So this would be like putting it into some distilled water, for example. Now what happens there is we've got a higher water concentration in our solution than inside our cell. So as a result of that, water moves from the area of high water concentration to the area of low water concentration, so it moves into the cells. That increases the turgor pressure of that cell, so it swells up. The middle one, we can see that what we've got there is we're placing our actual potato, for example, into a solution that's got the same concentration of water as inside. So because the concentrations are the same in the solution and in the cells, then the water moves in at the same rate that it leaves. So there's no change to the cell at all. If we then have a look at the diagram on the right, what we're doing there is we're placing our cells into a more concentrated solution. So this would be like a strong sugar or strong salt solution. And as a result of that, our cells have a higher water concentration than the solution. So as a result of that, the water is going to move from inside the cells to the surrounding solution, and that means we're going to lose the mass, because as water moves out, obviously mass decreases. Finally, we need to understand what happens to animal cells in those same 3 scenarios. So if we start on the left hand side first of all, where we're going to place our red blood cell into distilled water, for example, then because we've got a higher water concentration in our solution than inside our red blood cells, water moves into them. Now this is going to end up swelling our red blood cell, and because it has no cell wall, that means it's also likely to burst. If we think about the middle diagram, we can see that we've put our actual red blood cells in a solution which has the same concentration of water as inside them, and as a result of that we see the same amount of water entering the cell as leaves the cell, so there's no change to our cell at all. And finally on the right hand side there, we've put it into a very concentrated solution. So this might be a strong sugar or a strong salt solution, and what we're going to end up seeing there is, because we've got a higher water concentration inside the red blood cell than in the surrounding solution, water moves from inside the red blood cell to the surrounding solutions, and it then shrivels up, as we can see.